A gente vai receber agora o professor Justin Kinney. Ele já está com a gente, so I'm starting to speak in English because he's American. And we're receiving now Professor Justin Kinney, and he completed his PhD in psychology and neuroscience at Temple University, and he carried out his postdoctoral studies at the School of Biological Science in University of Southampton. And now he is an assistant professor at Wayne State University, United States, where he focuses on understanding the molecular and neural basis of individual differences in behavior, using the zebrafish as a model and applying behavioral analysis, molecular manipulations, and brain activity mapping. So, welcome, Dr. Justin Kinney. I hope you're seeing me.
Great, thank you. Can you hear me okay?
As well. You are free to start and share your screen with us.
All right, can everybody hear me okay? Yes. So just want to make sure everyone can hear me before I start. There's a nod of a head. Test, test. I want to make sure everyone can hear me before I start. There's a nod of a head.
You can hear me now.
You can hear me now? Yeah? Okay, cool. Thank you. Awesome. Well, thank you so much for the kind introduction. I really appreciate it. We're good? Okay. And thank you so much for the invite. I'm really excited to tell everyone about the work we're doing in my lab, and I'm saddened that I couldn't be there in person, unfortunately, due to COVID-related issues. I'm not able to travel at the minute, but I hope to one day in not too distant future to come and visit the beautiful country of Brazil. But anyway, so again, thank you so much for the invite. I'm excited to tell you about the work we're doing in my lab at Wayne State University, where we're studying the neural and molecular basis for individual differences in behavior using zebrafish as a model. Let's see. Okay, so first I just want to start with defining what I mean by individual differences. Oftentimes this is referred to as temperament or personality, and operationally this means behaviors that are consistent across time and context. In humans, individual differences or personality is often measured using the Big Five personality test, where people are measured on different traits such as extroversion, agreeableness, openness, things like that. And this is important because it helps predict things like disease risk, the likelihood that an individual might engage in risky behaviors, or stress management. Now, in animals, we can't do exactly the same thing as we do in humans with the Big Five personality test. They're not very good at self-report. So we have other ways of measuring behavior and individual differences. And this is looking at how animals respond to novelty, their fear responses, how they learn, different social behaviors, activity levels, etc. And we use this to model human behavior, but it's not just a model of human behavior and human personality. Individual differences in animals are thought to be important in their own right for contributing to evolutionary fitness. And this is not just of interest to the scientific community. Given the proliferation of all the different sorts of tests you can take for different types of personality out there, we can see this has also seeped well into the popular consciousness, where you can ask and answer questions about what type of Disney character you are, which Game of Thrones character you are, depending on your age. You might have kids wanting to know which Paw Patrol pup they are. So the idea of individual differences is prevalent in our society. Now, when we think about how individual differences come about, we, of course, we're studying the behavior. And the behavior is a manifestation of an interaction between biology and the environment. So, of course, different environmental conditions will elicit different behaviors. And different aspects of biology also contribute to individual differences in behavior. And the focus of my work is on the biology of individual differences. Now, when we think about biology, you can divide it into two different aspects. There's the shared biology, which is shared amongst all members of a species, for example. And then there's the unique biology. So individual differences in gene expression or other aspects of biology that might contribute to behavior. And as I mentioned on the last slide, these individual differences in the biology provide grist for evolutionary adaptation to a changing environment. So our environments are constantly changing. And so having a collection of animals that might respond differently to different environmental conditions when different types of behavior might be more adaptive than others is useful, at least at the species level. Now, we know a fair bit about the basic biology of individual differences and what aspects of biology contribute to individual differences in behavior. So, for example, it's well established that there are biological contributions to personality. So genetic influence on personality traits has moderate heritability on the order of 30 to 40% or so. However, it's highly polygenic. So many genes make small contributions to individual differences in behavior, which makes it difficult and challenging to study, especially in a human population. This means you need large numbers of people to be able to detect these differences. And of course, you cannot necessarily go in and manipulate the genes that you might find to be associated with personality. We also know individual differences in behavior. Genetics contributes to that in animals as well. The most obvious that I think many of us are probably familiar with is strain differences. So we have different strains of fish or other different strains of mice or other model organisms. And we know they have differences in aggressiveness, learning, social hierarchy, etc. That you can selectively breed animals based on their individual differences in behavior, and you can enrich those behaviors in those generations. It can often occur within 1 or 2 generations, suggesting there's a strong genetic component. And this is particularly obvious if we think about wolves and dogs who are descended from wolves, and how selective breeding of dogs is not only gives us dogs that have different physical characteristics, but also different behavioral characteristics. No self-respecting wolf would allow you to put them in a jean jacket, where there are several dogs strains that would allow you to do that, or dog breeds. From an application perspective, so thinking about how we can apply the idea of individual differences in behavior to medicine and health, one suggestion has been in the context of psychiatry and getting away from an approach to psychiatry that has a sort of a one-size-fits-all approach, where you use a trial and error approach to figuring out which sorts of treatments might work for any one individual. And oftentimes, the first thing you try doesn't fit particularly well. And there's a movement towards using a precision medicine approach instead, where if you can find the right things to measure, you can then tailor your particular treatment to the individual. And this might mean that depending on the measurements you make, one antidepressant or another antidepressant drug might be more effective, or might be able to determine whether psychological intervention might be more effective for a psychiatric disorder or other types of approaches. So this study of individual differences and the biological basis for it is not only relevant for understanding animals and their behavior and personality, but may have some application with respect to psychiatry as well. And so the animal organism, or the animal model that we used to study individual differences is the zebrafish. And in particular, we focus on the adult zebrafish. So why adult zebrafish? Well, adult zebrafish have a large behavioral repertoire on par with mammals. They show different types of learning, they have a complex social environment, and they do different types of exploratory behavior. Of particular benefit, they're small and relatively inexpensive compared to more commonly used rodent models, meaning that they're more amenable to medium and high throughput screens. And this is important because when you want to study individual differences in behavior, you need large numbers of observations so you can identify the presence of different clusters. You can't do this with 10, 20, 30 animals. You need hundreds of animals. They also have a relatively simple morphology for tracking, and this makes it easier to track fish as they swim around the tank and to be able to fully capture all their behavior. Genetic manipulation of zebrafish is fairly straightforward due to the fact that they have externally fertilized eggs. And despite being fish, they still retain a reasonable level of similarity to humans. They are vertebrates like humans. 70% of human genes have an obvious ortholog in fish, so oftentimes when a gene is uncovered in a fish, you can also find a similar gene in a human. And behaviorally, they're diurnal, they're highly social, and they use vision as a primary sense. And I and many in the audience aren't the only ones to have started to appreciate the power of the zebrafish model. You can see over the past 20 years or so, there's been a rapid increase in the number of publications using zebrafish. However, it still pales in comparison to all the publications using mice and rats, but it's increasing. This is a little yellow one down here. This is looking at the non-traditional model organisms, non-traditional, so not the mice and the rats. And zebrafish have been increasing, at least since 2019. I need to update this. So that's sort of my introduction, and just to give you an outline of the things I'm going to tell you about from my lab today. The first thing I'm going to tell you about is one example of how we've been studying individual differences in zebrafish behavior by looking at exploration of a novel environment, so a very commonly used task in zebrafish. I'll tell you a little bit about our progress in identifying the molecular basis for individual differences, particularly with respect to serotonin. And then finally, I'll tell you about our approach to identifying the neural basis for individual differences to develop, to capture patterns of whole brain activity. And finally, one point I want to emphasize is that the tools we use in our research, I make a point of making sure we use free open source tools. So these tools are free as in beer, meaning that they are free to use. You don't have to pay to use them. They're also free as in speech. So many of these tools you can manipulate and modify yourself. They're written in open source code like Python or R, things like that. So the behavior I'm going to tell you about today is the exploration of a novel tank, or the novel tank test. Now, the traditional way this has been done is a fish is put into a novel tank, and the video is recorded from the side. And you can generate a trace of the fish swimming around this tank. And the exploratory measures that are typically captured when an animal is placed into a novel tank is time in the top. So the time an animal spends in the top is thought to be a measure of boldness or approach, since being in the top of the tank is thought to expose animals to greater risk of predation by birds in their natural environment. And then people typically capture the distance traveled, so how far the fish swim while they're in the tank. And if you want to identify individual differences in behavior, a common approach is to do a median split. So you have some measure, like time in the top. And then based on the time in the top, you do a median split, and you say the fish on that are one side are high on this measure, maybe these are your bold fish. And then the fish on the other side, you might call your shy fish, which are low in this measure. However, when we were thinking about studying individual differences in the lab, we realized this approach had several significant limitations. One is that oftentimes only a small number of animals are used, so it's hard to know if there's more potential individual differences than what would be uncovered through a median split, which just uncovers 2. And the analysis is typically restricted to 2 dimensions. So fish are typically recorded from the side, so you can only get this sort of this XY dimension. But in reality, and in the wild, fish explore in 3 dimensions. So we wanted to expand this to identify and capture zebrafish behavior in 3 dimensions, which would then allow us to capture more different types of exploratory behavior. So hopefully this video is coming through. But what we did, the first thing that we did when I started my lab back in 2018, was to come up with a way to measure 3D swim behavior in zebrafish. Luckily, at the time that I started my lab, Intel released these cameras called RealSense depth sensing cameras. These are really cool cameras, not super expensive, a couple $100. And what they have on board is they have a typical RGB camera, which you can see here. And then they also have 2 infrared cameras separated by a small distance. And it uses the stereoscopic images from these infrared cameras, the disparity between these 2 cameras, to measure the depth in the image. You can see an example here of a fish swimming around our novel tank. This is the RGB camera, just like a webcam. And then this is the depth camera, where the color indicates how far away the objects are from the camera. And if you look closely, you can see the fish swimming around over here. Now, to track the fish, we use an open source tool that was also developed and released in 2018 called DeepLabCut. And I think postdoc Barbara in my lab will be telling more about how to use DeepLabCut for your own research. So we use DeepLabCut to track 5 points in the fish. And then we take the tracking from the DeepLabCut in the color video, we combine it with the depth video, and we get 3-dimensional swim traces for all our fish. Now, from these 3-dimensional swim traces, we extract 4 behavioral parameters. Like you saw on the last slide, we do distance from bottom. We also do distance from center, This is oftentimes referred to as stigmotaxis, so how far the fish is, how close the fish are to the periphery of the tank. We capture distance traveled, and then we also want to ask what percent of the tank the fish explores. So we divide the tank to 1,000 voxels and ask what percent of those voxels the fish explores in the tank. And this is work that was led by Neha Rajput. She's the first grad student that joined my lab, and she got an assist from a very precocious high school student, Kush, in doing some of the analysis. And like I said before, one of the other limitations of a lot of prior work is that they only got data from a small number of animals. We got data from over 400 animals here from 4 different inbred strains and both sexes of fish to make sure we capture as much behavioral variability as possible. So the first thing we did when we did this analysis is we just looked at the histograms or density plots of the different behaviors. And so that's what you see here. We see the different strains, AB, TL, TU, and WIC, and the different sexes, female and male, and the 4 different behaviors we captured: bottom distance, center distance, distance traveled, and percent explored. What we notice is that about 1/3 of the measures show non-normal distributions. So for example, here in AB males and percent explored, we see what looks like a bimodal distribution. In many of these, we see something that might be kind of normal, but there's also another mode over here. So we see a lot of complexity and non-normality in the distribution of these different behaviors. In addition, we don't really get many strong correlations between the behaviors. The strongest we get is about 18% of the variance explained, for example, between distance travel and percent explored. So that suggests that these 4 different behaviors that we're capturing here are capturing distinct elements of exploration. So the next thing we wanted to do is to determine whether or not there were the presence of different clusters of behavior in this data. And because we had this non-normal distribution, we wanted to turn to techniques that would allow us to capture the non-normality and not get confounded by it. And so the technique we turn to is a technique called building a k-nearest neighbor network and using clustering on this k-nearest neighbor network to identify clusters. So I stole this idea from the people that do single-cell RNA sequencing. I was looking at how they were doing clustering and saw this and thought that would also be a useful technique for us, and it turned out it was. So the way this works, the other reason I like it is I find it to be very intuitive. I really like graph theory and network analysis, and I find that a very intuitive way to understand clustering. So what you do here is you build a k-nearest neighbor network, where each node in the network, so each of these points here represents a node in the network, is connected to its k-nearest neighbors in 4-dimensional behavioral space. So this is behavioral space that includes bottom distance, center distance, distance traveled, and percent explored. And then we can identify network clusters on that network. So for example, in this little toy network we have here, where each point represents a fish, if k was equal to 4, we would connect this fish to these 4 fish that are closest to a behavioral space, this fish to those 4 fish, and so on and so forth. And then once we have a network, we can apply well-established network clustering algorithms like the Louvain algorithm to identify clusters. The other thing I like about this approach is that there's only one tunable parameter. So that makes it relatively straightforward to figure out what is the best clustering that we can get. And that tunable parameter is k. But now we need to determine, how do we choose k? How do we choose how many nearest neighbors we connect each individual fish in the network to? So there's not really an agreed upon way, as best I can tell, in the field about how to cluster. It's a very difficult problem that's not super well defined. So the best thing that we could come up with is that we chose k to maximize internal clustering metrics. So these are metrics that capture how well you've attached parts of the network to elements within a cluster versus between cluster. And we also want to choose k to be robust, meaning that if we make a small change in k, we don't get a big change in the clustering metrics, nor a big change in the number of clusters we identify. And so this is what we did, and this is what you see here, is that we varied k from about 10 to 200. Remember, we had about 400 fish in the network, or 400 fish that we were analyzing. And what we see is, if we look at, for example, this measure here, the Kolinsky-Harbaz index, where higher numbers here indicate better clustering, meaning you have more connections within a cluster than between clusters. We see that between around 100 and 125 or 130 and so, we have this regime where we have a lot of stability, meaning if we make a small change in k, we don't get a big change in this index. And we get pretty close to maximizing this index. We also looked at another index called the silhouette index. We get a similar result. and then the Davies-Baldwin index, where a lower number indicates better clustering, we also get a similar result. And so what we did is we chose the K in the middle of this, thinking that's our most robust K, so small changes don't make a big difference. And from this, we identified 4 different clusters. So when we take a look at the data, we do a dimensionality technique called UMAP, a uniform manifold approximation, kind of like a t-SNE plot, if you're familiar with that. And this is the data. This is all the 400 plus fish that we analyzed. We see each point represents a fish. The shape represents a sex, female or male. The color represents the strain. And then the bigger color on the outside represents a cluster. And so we get this nice clustering of our data set. If we take a look at the behaviors that are associated with each of these clusters, what's nice is that we captured, recapitulated a classic definition of bold behavior in this task, for at least when people use this to describe bold behavior. So these bold fish, we see they are higher than average for percent explored, so they explore a lot of the tank, and they're higher than average for bottom distance, so they're spending more time near the top of the tank than the bottom of the tank, demonstrating their willingness to take risk. We also have what we call shy fish. These are our fish that spend more time near the bottom of the tank and don't explore much of the tank. So in addition to this sort of bold and shy clusters, we also identified 2 other clusters. One we call active explorers. They're very similar to bold fish with respect to percent explored and distance from bottom, although not as high on distance from bottom. But the biggest difference is that they're swimming a lot more. So these are very active fish that are exploring the tank a lot. And you could argue that perhaps they're very inefficient at exploring because they're not exploring much more of the tank than our bold fish, but they are swimming a lot more. And then finally, we also identify a cluster we call wall huggers. These are animals that are average on everything except for center distance, spending their time near the periphery. I'm not 100% sure what the best interpretation is. This a shy behavior? Is this a bold behavior? But it certainly looks like it's a distinct behavior. Now, if we take a look at whether or not genetics contributes to this, so like I mentioned earlier on, it's been well established that genetics contributes to individual differences in behavior. What we see is that different strains and different sexes have different enrichment for different clusters. So for example, in our female AB fish, they appear to be enriched for wall hugging. And in our TL fish, irrespective of sex, these animals tend to be particularly shy. Notably, we have one strain, the Wicks, that don't seem to be enriched in any particular behavioral cluster. And the Wicks and the TUs, compared to ABs and TLs, as far as inbred strains, tend to have more genetic diversity, which might explain why, at least in the Wicks, we're not seeing any clustering. We're not seeing any overrepresentation. Now, the next aspect of individual differences is that you want to see some consistency across days. So with those 400 fish, what we did initially at the get-go, Neha put the fish in the tank on 2 days. And so we could ask if there's any overlap between the clusters on day 1 and the clusters on day 2. And indeed, we found that the fish, about 53% of the fish are in the same cluster on day 1 as they are on day 2, which is highly statistically significant. And if we look at how this varies based on the different clusters, we can see here on the diagonal, this is the fish staying in the same cluster. And you can see that's typically the most likely thing to happen. And as you can imagine, very few fish go from being in a non-shy cluster to a shy cluster on day 2. So on day 2, this is all the animals that are in the shy cluster. And animals that were shy on day 1 are more likely to be shy on day 2. But animals that are wall huggers, active explorers, or bold fish are highly unlikely to fall into cluster 1 on day 2. It's probably the most notable thing here. And even animals that were shy on day 1, they then tend to, only 40% of them remain shy on day 2, they tend to shift to other groups, suggesting there's some habituation going on. Now, we also wanted to ask if this is consistent over multiple days. So we put the fish in the tank for 5 days in a row, a subset of fish, TU fish, or every other week for 5 weeks. And what we see here, that's the data you can see over here. This is the daily data. This is the weekly data. About half the fish remain in the same cluster on 4 out of 5 days. So that's about here, the same color on 4 out of 5 days. That's also true over weeks. About half the fish are in the same cluster from over every single week for these 10 weeks of the study. What's also interesting from this is that we have a small subset of fish that change cluster every single day. We like to call these our consistently inconsistent fish, something that we've probably all experienced with people in our life who are consistently inconsistent. So maybe inconsistency in personality is a personality trait itself. But this also might suggest behavioral plasticity. So these are animals that are willing to try different behaviors in this novel environment. So there's different ways of interpreting that data. And if we look at the consistency over time during repeated exposures, what we find is that as we place the animals in the tank in subsequent days, you get higher and higher overlap. So for example, there's almost 80% overlap on the behavior on day 4 and the behavior on day 5, and about 75% overlap behavior on week 8 and behavior on week 10, suggesting there's some habituation going on and the animals kind of settle in to a behavioral response to the novel tank. And I think this is something we can all relate to. So the first time you go into an environment, so the first time, your first day at work, you might respond a little bit differently than your second day at work. And certainly by, you've been there for several weeks, you settled into a sort of behavioral response that you get into at work or any other novel environment. And so this makes intuitive sense. Now, the next thing we wanted to start asking is what's the molecular basis for this? So what is causing the animals to behave in one way versus another? And so we're just getting started on this, but I'll tell you about one story that we've started to develop in the lab, where we've looked at some neuromodulators and in particular monoamines in the brain and their contribution to individual differences in behavior. And the reason to look at monoamines is that, for example, in the human brain, there's only a small number of nuclei in which you get clusters of these cells that express, for example, 5-HT, that release 5-HT, serotonin, or dopamine, that then project to the rest of the brain. And these neurons are thought to help regulate behavioral state, how an individual would respond to environmental stimuli. In zebrafish, several different nuclei for serotonin and dopamine have also been identified. There's some overlap with humans and mammals, but not entire overlap. There are also some unique aspects to it. But they also project to multiple parts of the brain, suggesting these neuromodulators in zebrafish might be doing something similar to those in mammals. And so we wanted to study these monoamines and ask if there's some relationship between monoamine levels and the differences in behavior that we've seen in our task. And studying monoamines in the context of personality is nothing new. This has been done for at least several decades. So for example, in humans, this plot here shows the number of publications studying personality and dopamine or personality and serotonin genes since 1967. And you can see there's a big increase here, suggesting a strong interest in understanding how dopamine and serotonin, for example, contribute to personality. This is also true in animals, where boldness and aggression in animals, such as fish, birds, and rodents, have been associated with different monoamines. And this is an example from the zebrafish world, from Kai Maximino, who has looked at the effects of an SSRI, a selective serotonin reuptake inhibitor, on behavior in the novel tank. And interestingly, what I really like about this finding is that he shows that the effect of fluoxetine, this SSRI, on behavior in this tank can depend on the background strain of the animal, suggesting there's some genetic contribution to the role of monoamines in behavior, and that depending on the background, you can get different effects on altering monoamine function. So if we go to work and the way that we try to determine whether or not monoamines contributed to what we call boldness, the bold behavior that we saw in the tank, we developed this experiment, which was led by Fatima Bieglu, who is a visiting scholar from Iran in the lab, in collaboration with Shane Perrine's lab and his postdoc Cameron Davidson, which you can see here, who's got the great mustache. I need to grow mine out a little longer so I can twirl it like him. And so the experiment we did is that we placed animals into the tank 3 days in a row. We let the animals rest, and on the 5th day, we euthanized the animals, removed their whole brains, and subjected them to high-performance liquid chromatography to identify dopamine, serotonin, norepinephrine, and then the dopaminergic metabolite, DOPAC, and the serotonergic metabolite, 5-HIAA. And the reason we designed the experiment like this is that we wanted to get some measure of baseline levels of these monoamines, thinking that it's the baseline level that's going to be important for how animals respond to the 6 minutes in which they're placed into the novel tank. And to capture boldness, we developed this, what we call a boldness index, which is basically just the z-score for bottom distance and the z-score for percent explored combined. And this sort of captures the idea that the bold animals, that cluster 4 that we previously identified, was primarily identified and differentiated based on bottom distance and percent explored. So when we first did this, we briefly looked at whether there were any sex differences in behavior and brain chemistry, and we found several sex differences in how the animals behave over the 3 days of the task and in their brain neurochemistry. So we decided to analyze the female and male fish separately. And so when we look at correlations between boldness, so this is the boldness index, and brain chemistry, what we see is that the strongest correlation that comes out is this 5-HIAA 5-HT ratio. So this is a measure of serotonin turnover. And we get this nice medium-sized correlation in female fish, about 0.37, that we don't get in male fish on this first day of exposure to the novel tank. And this is when the novel tank itself is still novel, right? Every day you put the animal in, the tank is no longer novel. And so we focus on this, hypothesizing then that serotonin might play a role in individual differences in boldness in female fish. So to test whether or not this correlation actually led to some sort of functional difference in the serotonergic system in zebrafish, we designed another experiment where initially what we did is we placed the animals in a novel tank. And on day one, we did this, separated them by sex. Then we calculated their boldness index within sex, and then did a median split to identify shy fish and bold fish based on this boldness index. And then we asked whether or not the bold and shy fish would have different responses to a selective serotonin reuptake inhibitor. So this is a compound that would increase the amount of serotonin at the synapse. And so we're asking, does the boldness that we identify on day one affect the response to an SSRI, in this case, a drug escitalopram, on day two? So we give them the drug, and then 30 minutes later, we put them into the tank and ask if we have an effect that differs based on the boldness of the fish. Indeed, what we found in female fish, we get a really nice, strong interaction between the boldness of the fish on day one and whether or not they were given the drug escitalopram on day two. So we can see here that bold female fish get more bold. They spend more time near the center of the tank when given the drug, whereas shy fish actually become more shy when you give them the drug. And so we get this difference in how the animals respond to the SSRI, to escitalopram, based on their behavior on day one, suggesting there's differences in the way the serotonergic system is functioning in bold and shy fish. So this difference that we see at the neurochemical level manifests at the behavioral level as well. And we don't see this in male fish. We don't see any interaction between boldness and drug in male fish, which mirrors what we saw with the HPLC data, where we see this difference in female fish and not in male fish. Now, one thing I just want to briefly mention with respect to technique development is that, well, maybe I'll skip this part because I think this is only meant to be 40 minutes. So I'll skip this part and go on to the last part so I can talk about the neural basis of behavior. I don't want to run out of time too much. I'll just give you the punchline here. And the punchline here is that we developed, the way we gave the drug in our last experiment was based on using a new gelatin-based feed that we developed for drug administration in fish that allows you to give drug based on the weight of the fish. And I'll just give you an example, a brief example here of what this gelatin-based feed looks like and how it works with the fish. So this is the gelatin feed you can see here at the end of this forceps and the end of here. The end of the forceps here, and you can see the fish here, and I'll just play this video. And you can see these fish, they love the gelatin feed. And we see as you give the gelatin feed over subsequent days, you start to see a decrease in the speed in which they eat the feed. And within 2 to 3 days, they eat the feed within about 30 seconds. Okay, so just to summarize that part of the talk before I move on to the next part, we identified exploratory behaviors that fall into 4 different clusters: bold, shy, wall huggers, and active explorers. That's consistent across days and weeks. We see that 5-HT, serotonin, contributes to boldness, in female fish at least. And I only briefly described this gelatin-based feed for drug delivery, which is a nice, non-invasive, inexpensive, precise approach to drug delivery. Now, the next part I just want to briefly go over is how we're working to understand the neural basis for individual differences in behavior. And so the idea here is that we have these individual differences in behavior and these behavioral clusters we identified. And we want to ask, what is the differences in patterns of brain activity that result due to these differences in behavior? So we can identify the brain regions that might be differentially active in animals that are bold versus shy, for example. Now, I was developing these ideas as a postdoc when I was at Sick Kids Hospital. I realized to do this, we needed a brain atlas. In particular, we needed a digital brain atlas. And digital brain atlases existed for humans, of course. Mice, we have the Allen Brain Institute that makes a digital brain atlas. Larval zebrafish, several labs have created brain atlases for larval zebrafish. But at the time, for adult zebrafish, the only atlas that existed was in book form. And this was published by Mario Willeman back in 1996. And the reason we want a digital brain atlas is because it allows us to automatically segment a brain region into its individual parts, which is much less biased than having an individual go in and having grad students identifying and delineating parts. It's also much higher throughput, because if you want to do all the brain regions, it's not really feasible by an individual. And it also enables exploratory analysis, meaning that you don't have to have an a priori hypothesis about which brain regions might be involved. You can explore all the brain regions. And so to do this, we developed what we call ASBA, the Adult Zebrafish Brain Atlas. We did this by combining tissue clearing, so making the tissue optically transparent, followed by large volume imaging using light sheet microscopy, 3D volume generation, and then image registration and averaging before segmentation. So I'll go through each of these steps briefly. So for histology, we use tissue clearing. And the reason we did this is because if you take a chunk of tissue, brain tissue, it's optically opaque. And we wanted to get away from slice-based techniques because they're very slow, labor-intensive. You might lose a slice, and they can cause deformities. And so in the past 10 years or so, several groups have come up with tissue clearing techniques that can take a opaque piece of tissue and make it optically transparent. And the technique that we chose to use is called iDisco, which is fairly simple, takes 7 to 14 days, and works just about every time. Now, if you have a chunk of tissue, you have another challenge. The challenge is, how do you image a large chunk of tissue like a zebrafish brain that might be 1.5 millimeters thick and several millimeters long? And so to this, we turn to another technique that's come on in the past 10 years, the first commercial light sheet microscopes. These are microscopes that allow rapid large volume imaging and reduce photo bleaching. Here's an example of one here. And the principle here is that you illuminate your sample with a sheet of laser light from the side, and then you image in an orthogonal plane to that. And this way you can capture an entire XY plane of your image at once. You move your sample through the sheet, and within an hour or so, you can get an entire brain image of a adult zebrafish. These are some examples of the images. Hopefully these come through. I know it's a little hard sometimes seeing these sorts of things come through over these connections. But you see here, this is the original Willeman Atlas, a slice from that. When we took these images, we got autofluorescence images, and we did a nuclear stain. And the reason we did a nuclear stain is to match the crescent violet stain in the Willeman Atlas to help with segmentation down the road. So you can see these examples here, this nuclear stain. We're imaging in the horizontal plane, going from the most dorsal portion of the brain down to the most ventral portion of the brain. Now, when we did this, we wanted to get away from segmenting an individual animal's brain. We wanted to segment an average brain in case there's some idiosyncrasy with some individual animal. So we use this technique called image registration. We generate an anatomical average. The idea here is that each brain that you get, you're going to capture in its native space, and there might be different sizes, different orientations, because you can't mount them all exactly the same. And then you know that they're essentially the same. So you can register them into the same space. So you can change their orientation and their sizes to get them in the same space. Then you average them together, and you get an average brain. And it's this average brain we want to use for segmentation because it represents an average of multiple animals. So we had about 13 animals for this. And this is work I did with Patrick Stedman when I was at Sick Kids Hospital, initially at least. In addition to the staining I showed you, we also did some immunostaining for things like tyrosine hydroxylase. So this is an enzyme that's important for norepinephrine and dopamine synthesis, a neuronal marker, radioglial cell marker, serotonin, calbindin, and several others. And so we wanted to also use these not just to identify different parts of the zebrafish brain, but to help us segment different areas of the brain, because these different markers are enriched in different areas of the brain as well. And so to bring those images, right, we have those immunostained images that need to be brought into the same space as our nuclear stain, and these are different brains. And so the benefit of having this autofluorescence channel is that you get the autofluorescence image no matter what you do. And so we can take this autofluorescence image that's common to our stains and our topro, and we can apply image registration to these. And the transformation that we discover that registers one of these brains to the next, we then apply to the individual stains, and that brings the stains into the same space as the atlas. And now we can have multiple images all in the same orientation and space. And so we had these images. This part took several years, and we finally published it a couple of years ago now in eLife, where we segmented over 200 anatomical regions. And this took the collective effort of several undergraduates here at Wayne State, and including a collaboration with Thomas Mueller, who's out at Montclair State University. And this is an example of the segmentation you can see here in 2 dimensions. You can see here in 3 dimensions. And you're also welcome to go and you can explore the Brain Atlas. It's free up online. I have a great undergrad here, Chris Covert, who turned this into a website. You can go to asba.wayne.edu and you can explore the atlas. And if you want to explore it in more depth, you can download the individual files and use some free open source software to explore it in more depth. And here is a fun video, because why not? If you're going to make an atlas, you might as well make fun videos of the atlas. I hope the sound comes through, but it's not too loud. All right, so hopefully that came through and I got a smile out of you. My son, when I did this, he was about 5 at the time. He watched this like a million times. It was so much fun to put together. So this is something fun we got to do in the lab, is choose the music for this. Anyway, so now we want to link the behavior to brain activity. So this is the last part of my talk. And so the way we do this is using this approach using immediate early genes. These are biochemical markers of neural activity. So the fish, the idea here is that the fish, they swim and they behave. Neurons fire in the brain. This initiates the transcription of a certain subset of genes. The most famous one is CFOS or FOS. And then we can detect FOS mRNA or FOS protein, the neurons that were active when the fish was engaged in the behavior. And to do this, we turn to another relatively new technique called in situ hybridization chain reaction. This uses small 150 base pair DNA probes that then can bind to the mRNA, and we can attach fluorophores to so we can identify it. And this is an example of a zebrafish brain in which we've identified CFOS expression in the brain of the animal after the novel tank test. Now the challenge comes if you want to map brain activity in an adult zebrafish brain, is you have thousands and thousands and thousands of cells, hundreds of thousands of cells that you need to count. And of course, that again cannot be done manually. And so luckily, a couple of years ago, a group at University College London in England and the Technical University of Munich in Germany came up with this suite of Python-based software tools for computational neuroanatomy called BrainGlobe. And one part of this BrainGlobe suite is called CellFinder. And notably, this is all done in Python. It's all free, open source, available online. So you can go and download it and you can go to the website. And the way this works is that you have a CFOS fluorescent channel, you have an autofluorescence channel that comes with it. This is just sort of background staining. And what you do is you train an artificial neural net to identify CFOS positive cells from artifacts. So for example, in our CFOS channel, sometimes we get bleed through of the autofluorescence into our CFOS channel that might get mistaken as a cell. But what this artificial neural net does is it compares this channel, the CFOS channel, to the autofluorescence channel. If it's present in both, then it says, okay, that's a non-cell. And if it's only present in one, then it's a cell. And so to do this, because this is a machine learning algorithm, a supervised approach, what Neha did is she labeled 10,000 cells and 8,000 non-cells to train an artificial neural net to over 95% accuracy to identify these cells. So we now have that as of a couple months ago. And so the first thing we did is we asked, what's the time course for CFOS expression? So we exposed animals to the novel tank, we dissect out the brains, do in situ HCR plus iDisco to make the brains transparent, and light sheet microscopy to identify the activity in the brain of the animals following exposure to the novel tank. And what we see is that the number of CFOS neurons rapidly goes up within about 5 minutes and then comes down by about 60 minutes. And so we're choosing this 15-minute time point here for identifying patterns of brain activity following behavior in the novel tank. And so with these tools in hand, we're on the precipice. We haven't quite done it yet. We have a lot of the data. We're working our way through it, but we're very close. And I showed you the tools and techniques, the open source tools and techniques we're doing, so we can identify individual differences in patterns of in behavior and the neural basis for them. Okay, so that takes me to about the end of the talk. So just to summarize what I told you. I told you about one way we've gone about identifying individual differences in behavior, in particular exploratory behavior. One of the other projects we're working on in the lab is looking at individual differences in contextual fear memory expression. I told you about some molecular basis for individual differences that we've identified, so serotonin, and some future work we're doing to follow up and identify novel genes that might be involved is using RNA sequencing. So we've got that data and we're working our way through that. So keep a lookout for some really exciting work identifying novel genes and pathways that are involved in individual differences in behavior. And then finally, I showed you how we're going about to identify the neural basis for individual differences in behavior. We have this free open source tool, ASBA, you can use to explore the brains of zebrafish. Use another open source tool, BrainGlobe, to capture neural activity. And of course, our future work is to map the regions associated with behavioral clusters. And we're also working on ways to manipulate those brain regions. And then I'll leave you with one question for yourself. You can ask, what kind of fish are you? We can add this to our which Game of Thrones character are you? Are you a bold fish, a shy fish? Oops. Yeah. An active explorer, or a wall hugger? Okay, with that, I'd like to thank the folks that did all this work. The people's work I highlighted from my lab was Neha, Alexander, and Fatima. The collaboration with Shane and his postdoc Cameron. Other members of the lab that have the ones in italics have contributed to some of the work you've seen here. I started the Atlas at St. Kitts Hospital as a postdoc and finished it at Wayne State. Our collaborator, Thomas Mueller, our funding from the NIH. And you're welcome to check me out on Twitter. I guess I need to change this to X now, at Fish for Brains, or our website, kennylab.com. And I thank you, and I look forward to any questions.
Thank you, Justin, for your talk. It was awesome. I will ask the people here if they have any questions. So we will come back. Alguém tem alguma pergunta? Algum comentário? Just a second.
Hi, Justin, and thank you very much for your talk. It's always fantastic to see what you can do in zebrafish. I have two questions actually. The first one is that you said you work with 4 strains of zebrafish, right?
Mm-hmm.
And they were all raised in labs, or they were wild caught.
So they're all, oh, sorry, go ahead.
And they're all from the lab strain?
Yeah, so they're all raised in our own lab. We got the original parents from ZIRC, the Zebrafish International Resource Center at the University of Oregon. So they're all inbred strains. They're wild type inbred strains, but they're not sort of outbred sort of from the wild. So,
because it surprised me that even in these inbred strains, you still find 4 types of personalities, let's see, only in the axis of boldness, isn't it? Because it surprised me. So the variability is still kept despite years of inbreeding. Is this?
Yeah, so I mean, we do see some biases, right? For example, the AB fish seem to be wall huggers. But yeah, I think part of that might be because they're not fully inbred, right? So no one has a fully isogenic inbred fish line. And so that might be where some of the variability is coming out. But even so, before I worked with zebrafish, I did a lot of work with mice, working with C57 BL6 mice and 129 SVs and things. And even in those isogenic lines, you still get variability in behavior. So I think it's probably due to some interaction between the genetics and the environment, perhaps during development, things like that, that give rise to these individual differences, despite the fact that they might be highly similar genetically.
It's amazing. I wonder how it is in the wild, how variable it must be. Maybe it could be not 4 clusters, but more normal, no normal distributions of a trait instead of maybe. So my second question is that there was a break in your presentation while you were talking about that. So we had unfortunately missed it a little bit, but you said that 50% of the 53% of the fish were consistent, so 47% were not consistent, right?
Yeah, that's true.
Okay, so what it means is that when you did the brain analysis, did you do that in the first day, in the second day? So how do you, is it possible that this brain chemistry changed from one day to the other? We see that there are features that can change sex back and forth. So
yeah,
maybe the brain chemistry can change from one day to the other?
This is a great question, and it's something we have debated in the lab. We don't have the best answer for it, but what we've been moving towards is thinking of just looking at the response to novelty. I think the challenge becomes, right, once you put the fish in the tank on the second day, it's no longer a novel tank. It's now at least a partly familiar tank to some extent. So you really can't get the novelty twice in a row. And so we've been debating whether the best way to do this is, the reason we did the 3 days in a row, for example, in the HPLC experiments, is because we weren't sure if the brain chemistry would correlate with the novelty response or sort of the consistent response that we see on day 3. So I think your point is very well taken. And it's an active debate in the lab as to what the best way to do this is. But right now we are, for the brain mapping, we're just looking at the novelty response. So putting the fish into the tank after 1 day and asking about the individual differences in behavior on that first day, just to get the neural activity that underlies the specific behavioral response.
No. And maybe the wild type is the one that is consistently inconsistent.
I would love to say I think those are the most fascinating, but they're only a small proportion of the animals. So I think this idea of behavioral is good.
How many of them are these consistent, inconsistent? What is the percentage of this?
What was the question? Sorry, can you repeat it?
Of all the 400 animals, you said a part of them are consistently inconsistent. What is the percentage of individuals that are like that?
So yeah, we don't have that from, let me see if I can share my screen. We don't have that from the 400 animals. From the 400 animals, we only did that for 2 days. We took 50 animals and did that for 5 days. So, and so of those, I would say maybe 10% at most were consistently inconsistent. We haven't actually quantified it, but it's so small that it makes it really hard to study. So.
Okay, thank you very much.
Thank you for the wonderful. And just to your point about wild fish, I would love to get some wild fish in the lab. I'm just afraid of them bringing diseases into the lab. So, anyway.
Thank you. Hi, Justin.
Hey, Anna.
Nice to have you here.
Yes, good to see you.
I want to ask you something very technical about the gelatin feeding you're using there. So do you feed each fish individually?
Mm-hmm.
And how do they learn to feed from the pipette? And the other question is more philosophical, related to stress. So do you think very strong, stressful situations or very chronic stressful situations can change their personality, can change how they show themselves in novel environments or in any other place?
Yeah, so that's one of the reasons we developed the gelatin feed, is to get away from stressful manipulations of the drug. Now, not all fish will eat the feed from the forceps. That's just, Alexander was excited to see our fish actually, some of them will do that. Most of the time you put the gelatin feed on the top of the tank and you move away, and within about 5 or 10 seconds, the fish will come up and eat it. After habituation. Some fish take a little while the first time. So we usually have to have a couple days in which we just give a blank feed so the fish get used to eating it. And there just seem to be some strange differences. So the TL fish seem to do it very quickly, which we showed in the video, whereas the AB fish tend to be a little more neophobic, it seems. So yeah, it definitely takes a little bit of time and practice and being very careful with giving the feed. Did I answer your question, or did I miss?
Regarding the stress for a long time, and if it changes how the brain works and how personality is shown.
Yeah, we haven't looked at it, but I would imagine absolutely. If you wanted to give drugs over an extended period of time, and you were doing something where you stress the animals every single day to give the drug, then yeah, I think the gelatin feed would be superior for especially extended drug administration. I think that gets to your question, right?
Yeah. Okay, thank you.
Sure. Sorry.
Hi, Justin. I have a question for myself. As the professor said, we missed a little of a few minutes of your talk, but nothing to worry. I was wondering about the four characterizations of personality that you are bold, shy, and more active and less active, I think. So is that what is the linear, right? The threshold. What's the threshold between them for? Because here we used to work only with bold and shy, and you propose two more. So there are some kind of difference visually, or only when you analyze the data, you.
So your question is, how would you determine a bold fish from a shy fish, kind of?
And with the two,
And the other ones. Yeah, so this is, I think, a fundamental issue with clustering and categorization. There's always going to be edge cases, like fish that are, and this is true for human personality, right? Not everybody is going to be a blatant extrovert or a blatant introvert. You might have some extroverted or introverted, traits, for example. And so if you really wanted to just isolate the really bold fish from the really shy fish, you might, instead of doing a median split, would want to take animals that are like in the top quarter and the bottom quarter, something like that are clearly, bold and clearly shy. The problem is, of course, then you get rid of animals that you might not, you reduce your power. The other way to do this that we've done is that we put the fish in the tank for 3 days, for example, and then we look for animals that are consistent on 2 out of those 3 days at least. So we can say those fish are consistently bold, are consistently shy, to help deal with this fact that they might be like on the edge and bounce back and forth. So there's a couple of ways to deal with the issue, but fundamentally, it's a problem when you want to give a categorical description of something that might be more continuous, and you're always going to have these edge cases. So just thinking about, depends on your particular experimental question, how far away they really need to be in terms of behavior, how bold is bold and how shy is shy, for example. Does that help a little bit, or does it make sense? Okay.
Mais alguma pergunta? No? So we end this for now. Thank you, Justin, for your time and your awesome talk with hope we can see other time in the future. Thank you so much.
Yeah, thank you so much for having me. Thank you, everybody.
Are you there?
Yeah, I'm still here, but I'll leave the studio.
Thank you so much. Bye-bye.
Bye.
Televisão.